So these are the three types of white blood cells. Let's discuss these features, the features of these one by one. Initially, we have the monocytes. Monocytes are very large white blood cells, much larger than the red blood cells, and their shape of nucleus is bean shaped, and a large nucleus. You can see this large nucleus bean shaped and kidney shaped. The other cell neutrophils are also pretty large. They are also quite large, much large, way much larger than the red blood cells, and they have the nucleus which is a lobed nucleus. You can see multiple lobes here. And then you have another kind of cell which is the lymphocyte, which I just discussed. This is the lymphocyte. So when the lymphocyte is there, lymphocyte is such a small cell that it is even smaller than a typical red blood cell. The cell is filled up, the most of the volume of the cell actually is filled up with the nucleus. So what's an antigen? An antigen is a substance that is foreign to the body and it stimulates an immune response. An antibody is what we'll be discussing shortly. So an antibody is a glycoprotein, which we also refer to as an immunoglobulin. It is made by plasma cells which are derived from B lymphocytes. These are secreted in response to an antigen. The variable region of an antibody molecule is complementary in shape to its specific antigen. This is what the chapter of immunity or the topic of immunity basically deals with in detail. The immune response is a complex series of responses of the body to the entry of a foreign antigen. It involves the activity of lymphocytes and phagocytes, and we need to just follow how lymphocytes are helping out the phagocytes. Actually, they both are helping each other out to make sure that the antigen that got entered into the body, it gets cleared up. Non-self antigen refers to substance that the cell is recognizing by the immune system as a foreign structure which will stimulate an immune response, and self is your own. I think this is what we already covered. So what are antibodies? Antibodies initially, when they are produced, they are attached to the cell surface membrane of the B lymphocytes. And when they are attached to the cell surface membrane of the B lymphocyte, we call these antibodies as antibody receptors as well, or we simply call them as antibody molecules, which are attached to the membrane of the cells, which is the B lymphocyte. So you need to first of all keep in mind that there is a type of lymphocyte called B lymphocyte, and these B lymphocytes have antibodies attached on its plasma membrane. These B lymphocytes can be differentiated and they can form plasma cells. So plasma cells are derived from B lymphocytes. Once a plasma cell is formed, plasma cell releases the antibodies which initially were attached on this B cell. So these antibodies are released in large quantity and abundant quantity, and these are the antibodies that we'll be discussing now. So the antibody is like a structure. It is a glycoprotein. It resembles the English letter alphabet Y. So in this Y, you can see these short arms here. They are the antigen binding sites. So this antigen can bind to this part of the Y structured molecule or to this part. So there are two antigen binding sites on one antibody for the same antigen. And this long arm of this letter Y is the site for the attaching to the receptor. You can see that this is the same region, this long arm of letter Y. This is the same region with which it is attached to the cell membrane of this B lymphocyte. So once it is attached, or initially when it was attached, so this is where it was attached through, and once it is floating, so these are the floating antibodies. They will be present in all the body fluids. So once they attach to the antigen, they hold the antigen, and from this end, this side, this is a constant region of an antibody. This region goes and bind to the neutrophil or the phagocytes, and phagocytosis proceed. So here you can see these two cells. Which cells do you think these are? These are neutrophils. And how do we recognize these as neutrophil? This is the lobed nucleus. You can see the three lobes of this nucleus here. So this is the neutrophil, and these are the engulfed pathogens. These bacteria have been engulfed by this neutrophil, and these bacteria are like about to be engulfed. So you can see that the phagocytized bacteria are already there in these vesicles, whereas these are yet to be phagocytized. So you know that when phagocytosis occurs, so the pathogen gets cleared up by the action of the lysosomes. So what's actually happening? So this is actually what's happening. You have the pathogen coming in your body, and this is the pathogen. These are the antibodies which will respond, and they will attach to its surface. Once the antibodies have attached to the pathogen, together they both will be attracted towards the neutrophil, or you can see the neutrophil will be attracted towards them. The neutrophil will bind to the antibodies, and the antibodies are in turn binding to the antigen, which is the pathogen in this case. Phagocytosis occur. You can see that the cell membrane forms a mouth part here, and it engulfs this pathogen into a temporary vacuole. This temporary vacuole goes into the cell, and we call this as a phagosome. So here you have a phagosome. The phagosome then will fuse with the lysosome. The lysosome will have several enzymes, including the proteases and lipases and carbohydrases and all the biological molecule digesting enzymes, as well as the enzyme that can digest toxins, the degrade toxins. So the lysosome will fuse with the phagosome, forming a phagolysosome. So once this is formed, the pathogen will be like cleared altogether, it will be killed, and all the useful substances which are present here, they will be assimilated within the cytoplasm, and all the debris will go out and it will be exocytized from the cell. Here you can see the repetition of the same. So neutrophil undertaking this phagocytosis and killing and digesting of these pathogens. So this bacteria is being engulfed. You can see that there are two bacteria shown to you. Both of them are being engulfed. Eventually they both are engulfed. So this bacteria is being engulfed. This is being engulfed as well. The difference is that this bacteria is being engulfed by the neutrophil without any involvement of the antibody, whereas this bacteria is already having an antibody which marks this bacteria for phagocytosis. This constant region of the antibody will bind to the receptor here. Once the receptor forms a bond here, this antibody and the bacteria will be engulfed together. So what idea you need to keep in mind that neutrophils phagocytize irrespective the antibody is attached to the pathogen, or if the pathogen is not having an antibody, neutrophils are designed to phagocytize. So when they are carrying out phagocytosis, yes, with antibodies mark them, the pathogens will be phagocytized at a much faster rate than they otherwise would be undergoing phagocytosis random. When we're discussing phagocytes, so we have discussed basically three types of phagocytes. We have discussed neutrophils. Neutrophils are the main phagocytes. They are short-lived phagocytic cells. About 60% of all white cells are neutrophils. They're highly efficient in engulfing bacteria, either in blood or in the tissue fluid outside the capillary, and they survive only a few days. We have long-lived phagocytic cells, which are monocytes, and monocytes also carry out phagocytosis, but monocytes are more designed to turn into macrophages. Macrophages are the cells which leave the blood circulation and they lie and they wait in different organs of the body. So you will have macrophages in the lungs, you will have macrophages in the liver, in the organs which are having various tissues. So macrophages will become the part of those tissues, those organs. These macrophages are not traveling in blood, whereas all these cells were actually traveling in blood. So macrophages are a part of a tissue or an organ, and they permanently reside there. So they keep on collecting the rubbish or the you can say the pathogens or the debris which is being produced there because of the functioning of that organ. So all these three types of phagocytes that we have just covered, they all are being produced by the same kind of cell which was derived from the same stem cell. So the stem cell was in bone marrow. The stem cell would migrate to the lymph nodes, and once in the lymph node, it would either turn into a neutrophil or it would turn into a monocyte. The monocyte can then go on to become a macrophage. So we have covered more about the first type of cells, which are phagocytes. Now about the second type of white blood cells, which are lymphocytes. So lymphocytes are very important when we are considering their role in immune response. There are two types of lymphocytes, B lymphocytes and T lymphocytes. B lymphocytes are also called as B cells, and T lymphocytes are also referred to as T cells. Since B lymphocyte and T lymphocytes, both of them are type of white blood cells, they both are produced in the bone marrow, but the site of maturation is different. B lymphocytes go on to mature in the bone marrow, they stay within the bone marrow, they divide and produce in bone marrow, they mature in bone marrow. But T cells, they are produced in bone marrow, but then they migrate from bone marrow and they are collected in thymus. Thymus is a gland in the chest region below the sternum bone, and this is where the thymus will provide them with environment and the chemicals which are required for the appropriate functioning of these cells. So these T cells, they start to mature up once they end up in thymus. This cell here, you can see this is an electromicrograph of a contents of a plasma cell, and you can see that in this plasma cell you have an extensive rough ER. These are all rough ERs here, and they are in extensive quantity, and they would have a lot of Golgi vesicles and Golgi bodies as well. Why? Because plasma cell is constantly secreting antibodies at a very fast rate, and antibodies are glycoproteins, so they need a lot of protein synthesis assembly, which means ribosomes and rough ER and Golgi to support that functioning. Here you can see how the lymphocytes are produced. So this bone marrow stem cell in the bone marrow divide by mitosis. It would produce a cell which will become another stem cell, and one cell will become a cell that can either turn into a bone marrow B cell, or it can go on to become a T cell, and during the T cell would migrate to thymus. This is the thymus below the sternum in the chest region, and this is where the T cell will mature up, and when it matures up, it becomes a T cell which is matured, and the B cell is the one which got matured within the bone marrow only. That's it for now. Thank you so much.